Okay. Good afternoon, students, and welcome to another lesson of ELIM's online biology. So today we're going to look at the need for transportation. First of all, we are going to examine the problems with complexity. And we're going to start by looking at the implication of the surface area to volume ratio. When we look at unicellular organisms like the amoeba, for instance, these organisms are actually able to exchange materials within themselves and their environment by using simple diffusion. And the reason why simple diffusion is sufficient to provide all the materials required by these unicellular organisms is because they have a large surface area to volume ratio. Simply put, it simply means that the area around the cell is enough to cater to the needs of the internal space within the cell. On the other hand, when we look at more complex organisms like human beings or plants or frogs or what have you, simple diffusion is not enough to cater to the needs of all the cells. And one reason for this is simply because the cells within these complex living organisms have a low surface area to volume ratio. Now, let's look at number two. Diffusion is insufficient as a process of transport. I think I've already described that with number one. And when we look at number three, it says organs are situated at great distances from each other. You will agree with me, for instance, that when we talk about the liver, for instance, okay, and we talk about the distance between the liver and perhaps the large intestine, okay, there is some distance between these two organs. And so if there's going to be any kind of exchanges between these two organs, it means that we need a proper transport system to ensure that materials are moved from or between these two organs. Now, again, let's look at some of the materials that have been transported in animals. Number one, we have oxygen. And as far as oxygen is concerned, okay, oxygen is a gas. We take in oxygen. This oxygen is transported with the help of red blood cells and is moved all over the body because all of our cells require oxygen to carry out aerobic respiration. And it is because of aerobic respiration that we are able to produce as much ATP as possible to direct cellular activities. Carbon dioxide is also another gas. Now, in the case of carbon dioxide, this is a waste product and it needs to be moved out of the body as quickly as possible because it can become toxic. There is, or should I say, there are proper channels through which this carbon dioxide are moved out from where they are produced to where they will be released into the environment. Number three, we have urea, or simply put, nitrogenous compounds. All right. So these nitrogenous compounds, like urea, or other ones like creatinine, okay, are moved out from the body through the kidneys. Of course, we also have the skin, which serves as an excretory organ, moving out salts and water. So which brings us to number 4, we have excess salts, okay? So the excess salts could be sodium salts, they could also be potassium salts. Either way, these two compounds are moved out of the body through a transport system. Now let's look again at some of the materials that are transported in plants. We have food, manufactured food that is. So in terms of manufactured food, we could be talking about sucrose, okay? We could be talking about glucose or even starch. So depending on what the plant wants to achieve, these things are moved from where they are produced or where they are stored to where they are going to be utilized. Plants equally have excretory products. These products include carbon dioxide and water. And just as in human beings, where carbon dioxide is released as a waste product, okay, carbon dioxide is equally released as a waste product of aerobic respiration in plants. Of course, water as well can be released as waste product via the process of transpiration. Of course, we already know that in plants, quite a lot of their waste products are either stored or used. For instance, carbon dioxide can be used for photosynthesis, while the other ones, like your alkalides, your anthocyanins, okay, all those ones are stored. So let's look at number 3. Water absorbed from the soil is transported to the leaves for photosynthesis and other functions. The water that is absorbed from the soil is passed through a transport system consisting of the xylem, the tissues of the roots, as well as the root hairs or root hair cells. Number 4, other materials transported in plants include oxygen, nitrogenous waste, latex, amino acids, glucose, lipids, hormones, mineral salts. So I'm going to talk about hormones, for instance, which some of us might know as auxins, gibberellins, cytokinins, florigens. So many of these hormones are actually transported through the sap, either by diffusion or active transport. In any case, they are transported through a transport system, which involves the phloem tissues. And the same goes for glucose and amino acids. And next, we look at the media for transportation. And what do we mean by the media for transportation? We are talking about the materials that are responsible for moving products within the body, either the body of the plant or the body of the animal, from one point to another. We have cytoplasm in unicellular organisms. Like I said, simple diffusion is enough to cater for the nutrient requirements of these unicellular organisms, and is equally enough to cater for the excretion of whatever waste products are produced by these unicellular organisms. So the cytoplasm serves as that medium within these unicellular organisms through which the food materials are moved from one point to another, okay, through which even enzymes are moved from one point to the other. When we look at number 2, we have cell sap or latex in plants. Cell sap can be found in the phloem, and this would be the medium through which your amino acids, your sucrose, your auxins, or what have you, will be transported from one point to another. And then we have number 3, blood in animals. So as far as animals are concerned, many of us are already familiar with the fact that the medium of transport in animals is the blood. And it is within the blood that the food molecules, the gases, the hormones, nutrients, what have you, are dissolved and are moved from one point to another. Next, we have the lymph. While the blood is found in the circulatory system, the lymph is found in the lymphatic system. Okay, so number 5, intercellular fluid. You might see this written as tissue fluid. And the intercellular fluid is actually that fluid that bathes the tissues of the body. It is actually produced by the contents of the blood leaking through capillaries into spaces we refer to as tissue spaces. So the fluid contains nutrients, it contains hormones, it contains um, amino acids or what have you, the things that are required by the cells of the tissues. So let's look at the channel for transportation, okay? The diagrams we have in front of us right now, they represents the circulatory system as is found in the fish and the circulatory system as is found in mammals. So for the fish, we can easily see that we have 2 chambers here. 1, 2, okay? For the mammals, we have 4 chambers here. Now, for the fish, what we have here is a type of circulation we refer to as single circulation. And the reason why we use this terminology is simply because the blood passes through the heart once to complete a circuit. We can see that blood, okay, is being pumped by the ventricles to the gill capillaries where they are oxygenated. After oxygenation, they are transported to the different sections of the body where the oxygen within this blood is utilized by the various cells in the various tissues. The deoxygenated blood comes back to the atrium and the cycle repeats itself once again. In mammals, what we have is double circulation. And the reason why we use the terminology double circulation is simply because the blood passes through the heart twice to complete a circuit. We can easily see that we have a left side and a right side. So we'll talk more about this as time goes on. We have already talked about the fact that in mammals, what we have is double circulation. Now, that double circulation can still be further subdivided into pulmonary circulation and even systemic circulation. For pulmonary circulation, for instance, we are talking about the movement of the blood between the heart and the lungs. So we have the movement of blood between the lungs and the heart here, okay? In this particular diagram, you can see that this right side of the heart is pumping blood through these two blood vessels that we refer to as the pulmonary arteries. And these pulmonary arteries are taking the deoxygenated blood to the lungs, both the left and the right lungs. Now, when this blood is oxygenated, it returns back to the heart through the left side. And as you can see, okay, it is coming into the left side via the pulmonary veins. All right. So it leaves the heart to the lungs via the pulmonary arteries, and it comes back to the heart via the pulmonary veins. By the way, anytime you see the word pulmonary, you know that they are talking about the lungs. So next we look at systemic circulation. Like I said, we can divide the double circulation into pulmonary and systemic circulation. So as far as systemic circulation is concerned, we're talking about the movement of blood between the heart and the other parts of the body. Now, for the blood to actually move to the other parts of the body, right? First of all, it would have to be pumped by the left side of the heart via this artery here. This artery is referred to as the aorta. And the aorta, being the largest artery within the body, will supply the different organs within the body via some other subsidiary arteries. Now, when these different tissues or organs within the body have used the oxygenated blood that has been supplied by the aorta or the subsidiaries of the aorta, the deoxygenated blood will find its way back into the heart through the right side. So it could come back through this particular vein here, which we refer to as the inferior vena cava. Okay. Or it could be coming from the head and the neck regions through the superior vena cava. So we can also talk about circulation in terms of the open and the closed circulatory system. Now, the open circulatory system is something you would usually find in arthropods, like your insects. And we say it is open simply because the blood that is pumped from the heart, yes, it does pass through blood vessels, but the blood is emptied into a space that we refer to as the hemocoel. So it is this space called the hemocoel that interacts with the tissues of the body. Nutrients can be absorbed by the cells within this space, and waste products can be released into the hemocoel. Okay. This simply means that the blood actually makes direct contact with the tissues of the body. Unlike what we have in the closed circulatory system. In the closed circulatory system, we see that blood is passed or is pumped from the heart through blood vessels, all right? And these blood vessels basically differentiate into smaller blood vessels that we refer to as capillaries, all right? Now, one thing you would easily notice from this diagram is the fact that the blood never actually makes direct contact with the tissues of the body. So what happens is the fact that you would have contents of the blood leaking into the spaces where you have these tissues, what I call the tissue space. And when the contents of the blood leak into these spaces, they form what we call tissue fluid. So what happens is that the cells of these tissues or organs actually interact with the tissue fluid and not directly with the blood within the blood vessels. So the glucose, the amino acids, and the like could be absorbed from the tissue fluid, and then waste products will be released into the tissue fluid. Of course, the waste products will find their way through other channels back into the blood. Here we have the structure of the heart. And here, we more or less have a vertical section of the heart. First of all, the thing to notice should be this sac called the pericardium. And the pericardium actually consists of about 3 layers in total. It ensures that, for instance, while the heart is pumping, right, it doesn't affect the neighboring organs too much. Okay, it doesn't brush directly against the neighboring organs. And you could also say that it equally acts as a shock absorber. In any case, when you look within the heart, you have 4 chambers, okay? 2 chambers on the left and 2 chambers on the right. The chambers on the left include the left atrium and the left ventricle, and the chambers on the right include the right atrium and the right ventricle. Separating these two sides of the heart would be the septum that you see here within the middle. Now, let's look at both sides a little bit more. We will see that separating the upper chambers from the lower chambers, we have valves. On the left side, we have the mitral or the bicuspid valve. And on the right side, we have the tricuspid valve. Now, collectively, we call both of them the atrioventricular valves. All right. Now, let's go back to the left side of the heart. The heart is a muscular organ, that is true, but the left ventricle is the most muscular part of the heart, simply because it is pumping blood, 1, against gravity, and then 2, most importantly, it is pumping blood through a long distance. Why the right ventricle, for instance, may not be as muscular, or is actually not as muscular as the left ventricle, is because the right ventricle is only pumping blood to the lungs. As you can see from the diagram, the right ventricle only pumps blood to the lungs via the pulmonary artery, and the distance between the lungs and the heart is not really that great. Okay, and then finally, we need to talk about the different valves that we have here. We can see that within the aorta, we have the semilunar valves, or what we refer to as the aortic valves. And we also have this half moon-shaped structure, the semilunar valves, as well, within the pulmonary arteries, okay? And we can decide also to mention the fact that we have tendons connecting each valve to the walls of the ventricle. These tendons are referred to as the chordae tendineae. So let's look at the heart from the outside. We can already trace the left auricle from this region. That is the much of the left auricle as we can see. As far as the left ventricle is concerned, the left ventricle is this particular region here. Okay. The right auricle or the right atrium is this region that we have here. While with some degree of certainty, we know that the right ventricle is this particular region. Now we can see layers of fat all over the heart, and then we can also see the coronary arteries. Now, by the way, the coronary arteries are those arteries that supply the heart with oxygen and nutrients, because the heart in itself, yes, it does pump oxygenated blood to the other parts of the body, but the heart equally requires nutrients as well as oxygen. So the coronary arteries ensure that the cells of the heart equally have their own share of oxygen. If we look on top, we can see arcing above the heart the aorta, or if you like, the aortic arc. And then you can see some of the tributaries or some of the branches arising from the aorta and root other parts of the body. We can see the superior vena cava here, as well as the inferior vena cava here. We can see these two branches of the left pulmonary veins connected to the lung. And then we can also see these two branches of the right pulmonary veins. Okay. Moving on, we talk about the different types of blood vessels that we have in the circulatory system. And I'm going to start with the smallest of them all, which are the capillaries. The capillaries are actually 1 cell thick. Okay. And then they have these tiny gaps in between them through which, the contents of the plasma filter into the tissue spaces, producing the tissue fluid. The capillaries are actually the closest blood vessels to your body tissues. Next, we move on to the veins, and we can see that the vein has a very large lumen or space within it compared to the artery. Now, the veins, in terms of how thick they are, you can easily see that they are not as thick as the artery. And the reason, obviously, is because these are basically returning blood to the heart. They are not particularly pumping blood to different parts of the body. You can see the different layers common to the two blood vessels, that is the arteries and the veins, what we refer to as the tunica adventitia, the tunica media, as well as the tunica intima, or what you all know as endothelium. So let's make a comparison between arteries and veins. The arteries have a very thick elastic wall because they actually have more elastic fibers compared to the veins. So the veins are described as less elastic because they have less or a lower concentration of elastic fibers. The arteries, they are thicker, they are more muscular, okay, because apart from even having a higher concentration of elastic fibers, they have more collagen and they have more smooth muscles, okay, compared to the veins. The veins are thinner and not as muscular as the arteries. Then we have number 3, The arteries carry blood away from the heart. Okay, we already know that the veins return blood back to the heart. But there is a disclaimer, and the disclaimer is the fact that all arteries except the pulmonary artery carry oxygenated blood, while all veins except the pulmonary veins carry deoxygenated blood, right? Now, the color of blood carried by the arteries can be seen as bright red, while the color of blood, because blood in the veins is usually deoxygenated blood, and it appears dark red in color, not blue, actually. So, in any case, let's go back. In terms of the valves, because that's another important feature of both of them, arteries do not usually have valves, okay? But veins do. Okay? So, like I said, arteries don't usually have valves. In fact, the only artery that has anything close to a valve will be the aorta, okay? But veins, yes, the presence of valves are very, very unique to veins. In terms of the size of the lumen, from the diagram we looked at, we can see that the veins have a larger lumen compared to the artery. Now, when we look at the pressure within both blood vessels, we can see that the pressure within the arteries, okay, is much higher, while the pressure in the vein is much lower, as expected. So next, let's look at the transport medium within these blood vessels, at least as far as more complex organisms like human beings are concerned. All right. All right. So blood consists of a watery fluid called plasma and living cells called blood corpuscles. It is important for us to know that your blood is made up of cells, which we refer to as corpuscles, and it contains the liquid part, which we refer to as the plasma. So in terms of the cells, usually we talk about the red blood cells and we talk about the white blood cells. And in terms of the plasma, we talk about all the things that are dissolved in this liquid aspect of the blood, all right? Now, we're going to look at the cells in detail, but before we do that, it is imperative that at least we have at the back of our minds some examples of what we should expect to find in the plasma. And such examples would be plasma proteins, antibodies that's responsible for our immunity, enzymes, digested food, dissolved gases, salts, and waste products. So these are the kind of things that you would expect to see within the plasma, right? So let's go to the blood corpuscles or the blood cells, starting with the red blood cells or erythrocytes. As far as erythrocytes are concerned, they are described as being biconcave, all right? And being biconcave means, you see, they curve inwards on either side. There are about 5 million of them per cubic meter of blood. In terms of diameter, they are about 7 micron meters or 0.0072 millimeters in diameter. when they mature, they tend to lose their nuclei to give more space for hemoglobin. And yes, they are flexible. One of the reasons why they are flexible is for them to be able to squeeze through the narrow capillaries. So what are the functions? Your red blood cells contain hemoglobin, and it is the hemoglobin that actually binds to oxygen. And the release of this hemoglobin to other cells helps to facilitate aerobic respiration. Secondly, the red blood cells are actually formed from stem cells within the red bone marrow, as well as within the liver of the fetus. Thirdly, they have a lifespan of about three and a half to about 4 months. When they are worn out, they are destroyed in the spleen by phagocytes. And then the hemoglobin, okay, that is formed from the breakdown of these red blood cells. Remember that this hemoglobin is actually a combination of what we call the heme, okay, group, and the globin. The globin is the protein part of the hemoglobin, right? So while the protein part of the hemoglobin may be recycled or may be converted, broken down to amino acids, and from amino acids to urea, the heme group, okay, could equally follow I wouldn't say follow the same fate, but the heme group is that particular organic compound that is excreted as bilirubin, okay? And you may wonder what bilirubin is. Bilirubin is a yellow compound, and when you urinate, it's one of those things that actually makes your urine yellow. In fact, when somebody has a case of yellow fever, for instance, where you have quite a lot of the red blood cells being broken down, you have a release of hemoglobin, an excessive release of hemoglobin. And when this is broken down into heme and globin, part of the heme will obviously have to be excreted, but they would be converted, first of all, into bilirubin before being excreted via the kidneys. And like I said earlier, it's one of those things that actually makes your urine yellow. It can make the white of your eye yellow, and the bilirubin can equally find its way under your skin. All right. And those are some of those symptoms you notice when you have yellow fever. So let's look at the white blood cell. The white blood cells are not as numerous as the red blood cells. As you can see, we have about 7,000 per cubic meter of blood. If by any reason you go to the medical laboratory and they find out that your white blood cell count is up to 11, 12, 13,000, it simply means you have an infection somewhere, okay? So they are larger in size, and unlike the red blood cells, they actually have a nucleus, all right? Number 3, the white blood cells are also called leukocytes. It's one thing to note. Number 4, they equally are formed from stem cells in the bone marrow, and then you also have some of them being formed in the lymph nodes. Your white blood cells can be grouped into granulocytes and the agranulocytes, right? So your granulocytes could be your neutrophil, okay, your basophil, your eosinophils. Those are your granulocytes, and your agranulocytes are your lymphocytes. right? So like we said before, you can see the granulocytes here. And of course, from their appearance, you can easily see why we call them granulocytes, because they have a granular cytoplasm. The granules you see here are actually enzymes, really, that are involved in phagocytosis. But having said that, the major one that is implicated in phagocytosis is actually the neutrophil, which constitutes the largest percentage of the granulocytes, right? For the agranulocytes, those ones that have clear cytoplasm, like I said earlier, we have the lymphocytes. We also have what we call the monocytes. Now, remember that your lymphocytes include the B and the T lymphocytes. Lastly, of the corpuscles, we have the platelets, which are actually referred to as cell fragments. Now, this basically gives you an overview of what happens during the clotting process. There's a reason why we refer to it as the clotting cascade. And before I actually tell you the nitty-gritty of the clotting cascade, I should inform you, first of all, that it is the platelets that are involved in blood clotting. And the reason why we have blood clotting, apart from sealing up injuries to prevent entry of microorganisms, it also prevents loss of blood, which is not very good to the human body. So why do we call it a cascade? The reason why it is called a cascade is because you have the activation of a particular compound. Let's say, for instance, this is factor 12A. Okay. Now, once this factor 12 here is activated, it becomes factor 12A, and activated factor 12 or factor 12A activates this factor 11. By activating factor 11, it becomes factor 11A. So this continues until we have the activation of what? Prothrombin, or should I say the conversion of prothrombin to thrombin. Now, thrombin mediates the conversion of fibrinogen to soluble fibrin. And it is your soluble fibrin that actually forms a mesh, okay, preventing the, or should I say, sealing up the wound, preventing the loss of blood, trapping all kinds of things there to ensure that infections do not find their way into the blood. Now, let's look at how the blood actually interacts with the cells of the tissues, right? So when we talk about the interaction of blood with the cells of the tissues, we must remember that blood leaves the heart, first of all, via the arteries. And from the arteries, blood will flow into the arterioles, all right? Like smaller arteries. Now, the arterioles will differentiate into capillaries, okay? And capillaries, again, will fuse to form venules. Venules will drain into veins. So we should understand the sequence of events here. First, we have the artery. The artery branches into arterioles. Arterioles will differentiate into capillaries. Capillaries will fuse back again into venules, and venules will drain into veins. So what happens between these section, okay, these capillaries that are arising from the arterioles? Under high hydrostatic pressure, we have the contents of the plasma leaking or being forced into the tissue space, thereby forming tissue fluid. This happens under high hydrostatic pressure because normally the water concentration within the tissue space is higher than what we have in the blood in this particular region. Okay, so you do expect that water should actually be moving from the tissue fluid into the blood. But that doesn't happen because the force of the hydrostatic pressure is greater than the effect of the water gradient. As such, rather than water moving into the blood by osmosis, the contents of this plasma is forced into the tissue space, forming tissue fluid, okay, like I said, under high hydrostatic pressure. So what happens is the fact that things like glucose, salts, hormones, amino acids, that is small amino acids that are, or should I say small proteins, and, that are small enough to pass through the gaps within the capillaries will find their way into the tissue fluid. And so we have exchanges, glucose will be absorbed, salts will be absorbed, waste products will be released by the cells into the tissue fluid. This is what happens at the capillaries, branching directly from the arterioles. By the time we look at the capillaries that are much closer to the venules, what is going to be happening is that, the blood that is flowing into these capillaries is flowing at a high pressure. By the time it gets to the capillaries that are closer to the venules, the pressure is much lower. As such, osmosis takes preeminence here. So what happens is that water now moves into the blood in this particular region. Why? Because the water gradient or the water concentration gradient is greater than the hydrostatic pressure in this area. So we have water moving into the blood plasma in this particular region. We have, waste products, probably salts, moving back into, and even proteins that are small enough to move back into the blood. One thing I did not mention while I was explaining the exchanges that occur between the blood in these capillaries that are very close to the arteriole is the fact that oxygen in the red blood cells diffuse into the tissue fluid, not necessarily through the gaps, but through the membranes of the cells lining the capillaries. Okay, because the oxygen molecule is small enough to actually diffuse through those membranes. Okay, so they actually diffuse through the cell membranes into the tissue fluid, and as such, the cells of the tissues here can now have access to the oxygen and carry out their aerobic respiration. After this, we know that the next thing that is going to happen is that these cells are going to produce carbon dioxide, and as such, carbon dioxide will diffuse back into the blood in this area where we have capillaries that are very close to the venial end. Now, I did say that we have small proteins that can move back into the blood. We have some salts, waste products like carbon dioxide, nitrogenous waste, like urea or what have you, that can move back into the venule, or first of all through the capillaries before going into the venule. However, it is not all the proteins that will find their way back into the venule. Okay, some of these proteins will drain through the lymphatic vessels. Okay, so you can see here it is written lymph capillary, or you could just say lymphatic vessels. All right. So the other materials that do not drain back into the capillaries that are close to the venules here will eventually, all right, find their way back into, will find their way through the lymphatic vessels, and the lymphatic vessels will connect back with the circulatory system via the subclavian vein. So that gives you a simple overview of the interaction between the lymphatic system, as well as the circulatory system. Now, in terms of color, your tissue fluid contains, for the most part, a lot of things that you have in your blood, but they are clear in appearance. They are not red because your red blood cells do not find their way into the tissue fluid. So the mechanism of transport in higher plants. Of course, when we're talking about transport in higher plants, we are talking about the role of the transport medium, like the sap. We are talking about the role of the transport tissues, like the phloem and the xylem. And then we're also looking at what exactly are the products being transported within the sap or even within water in itself, all right? So generally, we know that we have food, carbon dioxide, water, oxygen, nitrogenous wastes, amino acids, glucose, oxins, and mineral salts. So in terms of transport of water and mineral salts, there are three things we need to hone in on. First of all is what we call root pressure. Second is capillary action. And third is transpiration pool. All these three factors or concepts that we're going to talk about are things that are associated with the transport of water and mineral salts via the xylem. So what really happens? If you look at this diagram here, you find that this diagram has to do with what happens in the root region. And what exactly happens in the root region? The root actually has root hair cells. And for water to move into the roots, it means that you must have more water in the soil compared to what you have in the root hair cells, so that osmosis can occur, which is the movement or diffusion of water from a region of higher concentration to a region of lower concentration through a semi-permeable membrane. That being said, it is not only water that's going to be transported in this manner. We are also going to have the transport of minerals. Minerals, most essentially, okay, are transported by active transport. And for just to remind you of what active transport is, it is the process by which materials are transported against a concentration gradient across a biological membrane, and the expenditure of energy in form of ATP is actually necessary to carry out this process. Okay. So we have, first of all, water moving into the root hair cells from the soil. From the root hair cells, the water would move into the cells of the cortex within the roots. Of course, the water will move from one cell to another until it ends up in the xylem within the roots. For instance, if I should, let me clarify by drawing this diagram. Okay. All right. So let's say this is the root hair cell. It absorbs water, and the water moves from one root hair cell to another down a concentration gradient. It ends up in the cells of the cortex here. From the cells of the cortex, it passes through the endodermis, and from the endodermis, it ends up in the xylem there. Okay. Now, while in the xylem, sometimes the root does pump ions into the roots to actually lower the concentration of water. Now, the idea of lowering the concentration of water here is to increase the concentration gradient between the water in the soil and the water in the root so that the root absorbs more water. Okay. That is what this whole idea of root pressure is all about. Okay. The cells of the root could pump in more ions to increase the water concentration gradient so that it takes in more water from the soil. By taking in more water from the soil, it increases the pressure of water within the root. Now, the next thing that happens is that the water travels up the stem through the xylem vessels. Okay, as you can see, the xylem vessels no longer have complete partitioning, okay, because as they mature, the cells of the xylem actually accumulate lignin, and the partitioning breaks down eventually, thereby allowing one continuous column through which water can be transported, right? So water moves up the xylem within the stem as we have it, right? And what are the factors responsible for the movement of water through the xylem within the stem? We talk about things like capillary action. We talk about things like the force of adhesion. We talk about things like the force of cohesion. And most importantly, we talk about transpiration pull. So let's start with capillary action. The capillary action has to do with the fact that the narrow diameter of the xylem vessel causes water to quickly rise up the xylem vessels. In terms of the force of adhesion, right? Now, water actually has hydrogen bonds, and the force of adhesion is created by the hydrogen bonds that exist between the water molecules and the walls of the xylem vessel. So it sort of enables the water to anchor itself, or the water molecules to anchor themselves on the walls of the xylem vessels. This same hydrogen, or should I say hydrogen bonds, all right, are responsible for the force of cohesion. And when we talk about the force of cohesion, we are basically saying that as the water moves up, they tend to drag each other. That is, as the water molecules move up the xylem vessels, they tend to drag each other up simply because of the many hydrogen bonds that exist within these water molecules. Finally, like I said, the most important force, or the strongest of all of them, is actually the transpiration pull. And for us to better understand the transpiration pull, we will have to talk about what happens within the leaf area. Now, this is a cross-section of the leaf area, right? We have the palisade mesophyll tissues here. Okay, we have the spongy mesophyll cells here. Okay, we have the lower epidermis, we have the stomata, we have the guard cells, we have the upper epidermis, we have the cuticle, and all that. So how does the structure of the leaf play a role in the movement of water within the xylem vessels in the stem? When the water actually gets to the leaf area, it actually gets here through the xylem. And what happens is that it soaks the walls of the palisade as well as the spongy mesophyll cells. from here, it diffuses, or should I say from here, it evaporates. The proper word to actually use in this context is evaporation. So it actually evaporates into the air spaces. Okay, by evaporating into the air spaces, it saturates the air spaces with moisture, thereby creating a concentration gradient between the water, the moisture or the water vapor in the air spaces, and the water within the environment. So because we have a concentration gradient between the moisture in the air spaces and the environment, we are now going to have diffusion of water from the air spaces into the environment. All right. Now, the fact that we continuously have this stream of water coming to the leaf, soaking the cell walls of the palisade and the spongy mesophyll, evaporating into the air spaces, diffusing through the stomata into the environment, okay, means that when we come back to the stem here, okay, there is a continuous loss of water at the top of the xylem within the stem, okay? And because there is a continuous loss of water at the top of the xylem within the stem, it means that the pressure of water here becomes lower than the pressure of water at the bottom. So this causes water to be pushed up in this direction. So what we've actually explained is the effect of transpiration, which creates a transpiration stream as indicated by these arrows. The transpiration stream ensures that there is continuous movement of water through the xylem vessels in the stem into the leaf and then out of the leaf. So we're going to stop here for today. On Saturday, we will continue from this particular aspect of the topic. Thank you.